Adrian, we are live.
Hi, everyone. Welcome to our Neuro Book Club. Today we're looking at The Spike by Mark Humphreys. I'm one of your hosts, Adrian Fairhold from the University of Washington. Over to Tim.
I'm your other host. My name is Tim Vogels. I'm at the ISD Austria, and we're very happy today to be hosting Mark Humphreys. Mark did his PhD in Sheffield and then did his postdoc in Sheffield. And I just learned actually that he was pulled back into academia after he tried to leave. He went and worked for a startup before being drawn back into academia. And that was actually a very lucky thing for the computational neuroscience community in England, because it's fair to say that he's probably the heart and soul of computational neuroscience, at least in the UK. He's been organizing bi-yearly, I think, Mark, meetings for computational neuroscience that have grown into quite a social affair, or that used to be a social affair before the pandemic hit us. Mark got his first position actually in France, in Paris, and then went back to the UK, again, lucky for the UK, to work as a professor in Manchester, and has then been recruited away to work in Nottingham, where he is now the chair for computational neuroscience, together with the other British Mark, that is Mark Van Rossum. So the two computational neuroscience Marks are now in Nottingham, making Nottingham look very good. He is here today to talk about his book, The Spike, and I'll let him do the introduction for the book himself. Mark, without further ado, the stage is yours.
Okay, thank you, Tim and Adrian. Thank you for the invitation. Right, yes, so The Spike. So the book itself was intended to be three journeys in one. So for you, the reader, the context is that you are being faced with a small dilemma. You're sat in some fairly nondescript office somewhere in the world, and you're a little bit hungry. It's the middle of the afternoon, and you notice right in front of you there is the final cookie left in the office tray of cookies that one of your co-workers has brought in, and you're faced with the dilemma of whether to nab that cookie just before your 3 o'clock meeting or not. And what the book is about then is everything that flashes through your brain in 2 seconds between glancing at that cookie and deciding to take it or not to give away the ending. And the first journey then is the journey of the information through the brain in the form of spikes, starting from when your eye falls on the cookie and you initiate the stream of photons hits your retina, and you initiate the spikes coming from your eyeball that are telling you there is a cookie and a box in front of you. And that journey then takes the reader through the various regions of visual cortex. It takes it through the object recognition pathways and the action visual pathways. Takes you through parietal cortex, prefrontal cortex. It takes us through deep down to the basal ganglia, back up to the motor cortex, and into the spine. So it's a journey through the brain from the spike's perspective, and you, the reader, following the spike, and me, the narrator, as we track that spike through the brain. And that's the first level journey. The second journey you're on is, as we're traveling through the brain, it's about learning more and more about neuroscience from the perspective of the spike level of how things are done. So it starts with what a spike is and why the brain uses spikes at all. And it takes on ever more complex ideas of how spikes work in groups and how they work, how the dynamics of the brain appear, and then how they code things and so on. And then the final journey that the book is taking you on is a journey of the cutting edge of the last 30 years of systems in computational neuroscience. For all the things we've been able to do with all these fabulous new techniques we have now, the optogenetics, large-scale calcium imaging and electrophysiology, chemogenetics, all the things that we've been able to play with in the last 20, 30 years have opened up huge new vistas of what it is that spikes are doing to show us much of the new things that we have learned. And of course, the book also reveals a lot of the things that we still don't know, which is part of the reason for taking the spikes view of the world. So, so obviously Tim and Adrian tasked me with reading you a chapter, which was slightly more challenging because also the idea of the book is it's a fairly strong narrative line from beginning to end. So I've chosen chapter 6. Chapter 6, because it's perhaps the most self-contained. Obviously then it's going to have a few callbacks in it to earlier ideas. But by the time we get to chapter 6, you, the reader, are somewhere deep deep into about to turn up into prefrontal cortex. And we've been following spikes up and down layers of cortex, so through layer 4 up to the superficial layers and down to the deep layers, and then diving down into the white matter and jumping from area to area to area of the cortex through the white matter. And what chapter 6 is about is asking you to think about what you've just seen on those jumps between every area, what it is in particular you haven't seen. Yeah. Cool. So to begin. All right, so chapter 6. So the dark neuron problem. The classic view. So every time we've exited the white matter, the view has been awe-inspiring. Like gazing at the night sky, all around us a panorama of scintillating neurons, points of light sparking with spikes. But just like the night sky, there is more darkness than light. Those bright points in the firmament of cortex are but pinpricks on a massive engulfing darkness. A darkness of neurons that are not firing. Wherever we follow our spike, there is darkness. Now closing in on the final regions of the cortex that deal with vision, where color is merged with shape, a light brown chocolate chip cookie, where curves are formed into faces, that Angela and Ishmael are at the office door, not looking this way. Our spike has zipped past billions of neurons in your cortex. The overwhelming majority of them have not fired a spike in the second we have traversed your brain so far. The overwhelming majority do not send anything. Even neuroscientists can find this hard to grasp, and little wonder, as our data have shown the opposite. Neuroimaging, functional MRI, shows us technicolor images of the cortex, its regions lit up in a swirling riot of poorly chosen colors that make the Pantone people cry into their tasteful coffee mugs. The swirling colors seem to show us that the cortex is a swarm of activity, that when we see a face, the visual areas of our cortex bloom with barrages of neural firing, from V1, V4, and down to the face areas of the temporal lobe. That when we hear a swell of strings, the auditory areas of our cortex bloom with barrages of spikes. Classic studies of single neurons seem to show us that each and every neuron has a role, that each responds to something, a line, a corner, a movement, a color. For when experimenters lower the thin sliver of an electrode into the cortex, they can readily record the spikes as they are dispatched from a neuron's body, and can relate those spikes to something happening in the world. Many have related these spikes to speeches of the visual world, like the neurons we have already met, the simple and complex, to the edges and lines and angles, the contrasts, shapes, objects, faces. Others we could have met had we taken a different starting point. Neurons in the auditory cortex that respond to sounds of a particular frequency. Neurons in somatosensory cortex that respond to touch on a finger, a toe, an arm. With decades of work, tens of thousands of studies showing us that when we lower an electrode into a bit of cortex, there they are, a swathe of neurons responding to the things they like. So surely all neurons send spikes. So some simple arithmetic will show why this makes no sense. Back when we were basking in the simple cell in V1, if we'd hung out a little longer, we would have seen it fires perhaps 5 spikes every second. And we know it needs about 100 spikes arriving at its excitatory synapses to make one new spike. So 5 spikes in a second means it needs a total of at least 500 excitatory spikes arriving in that second. But we also know that a V1 neuron has about 7,500 excitatory inputs. So if each of those inputs was sending at 5 spikes per second, there would be 50,000 input spikes in total every single second. And that's too many by a factor of 100. The simple cell in V1 should be sending 500 spikes every second. But it isn't, and neither are its inputs. And they can't. The sending 500 spikes every second is a neuron screaming at the top of its lungs. It's about the theoretical maximum rate a cortical neuron can produce spikes if forced to do so by an experimenter. Not least because after each spike, there is a few milliseconds in which a neuron can't make a new spike at all. So no, even the most active neuron in the cortex can only sustain a continuous output of about 30 spikes per second. So a paradox. The neurons in cortex are sending spikes at least an order of magnitude lower than they should be if all their inputs were similarly active. The only way out of this paradox seems to be that most inputs to a neuron in the cortex are not sending spikes. which implies that most neurons in the cortex are not sending spikes. Is this true? How to find a dark neuron. So up until the 1990s, neuroscientists could only record individual neurons in an animal blindly. They inserted their tiny sharp electrode into some bit of cortex and only knew they had found a neuron by the blip it made on their oscilloscope or the noise it made on the speakers in their lab. Tick, 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 tick. Which meant they could only find active neurons because their only way of finding neurons was by their activity. So this created a fearful bias in our understanding of spikes and which neurons sent them. If every neuron you record sends spikes, then as you are sampling at random, blindly, this implies every neuron is sending spikes. But if your only way of finding neurons is by finding spikes, then by definition, you could not find neurons that did not send spikes. Indeed, they would be dark matter, contributing mass to the brain, but invisible to all your instruments of measurement. But then neuron imaging came along. We point a digital video camera at a bit of brain, and in that bit of brain, each neuron contains a chemical we've injected that lights up when the neuron is active. Most often, that fluorescent chemical is responding to the amount of calcium in the neuron's body, lighting up to an influx of calcium with every spike. By filming that bit of brain in sharp focus, we can see all the neurons with our eyes, see their outlines, and we can see which ones light up. It turns out, for decades, we've only been recording the tip of the iceberg, but most neurons we see in these videos are not active. Our first clue to the scale of silence came from imaging the cortex of anesthetized rats. So under many anesthetics, the cortex operates just like it does in deep sleep, jumping between active and quiet periods about once a second. So imaging the first auditory and somatosensory touch bits of cortex, Jason Kerr and colleagues reported that the active periods actually contained only detectable spikes from just 10% of all neurons. So 90% were silent in each second or longer active period, and almost all totally silent in the quiet period. And this scale of silence wasn't because of the anesthesia. It is found in the cortex of behaving animals too. Christopher Harvey and colleagues in David Tank's lab at Princeton imaged a section of parietal cortex which lies at the end of Highway 2 in mice running inside a T-shaped maze. A T-shaped maze in virtual reality. The mouse ran on a ball while the virtual world moved around it. They reported that just 47% of neurons were active while the mice ran the maze. And even that low number required stretching the definition of active to its breaking point. A neuron was deemed active if it had more than 2 spike-like events in a whole minute, vastly longer, 10 times longer, than it took the mouse to run the length of the maze. And just in case you're thinking that putting mice in a virtual world may muck up their neural activity, rest assured. Researchers in Carol Saboda's lab have made an industry of imaging neurons in mice doing stuff in the real world. In a study led by Simon Perron, they imaged the specialized bit of the cortex that gets input from a whisker, while that whisker was being used to find a pole, and which the mouse wanted to find as it was thirsty and the location of the pole told it which spout would have water in it. So even in this special bit of whisker cortex that only cares about that one specific whisker, they found only 67% of neurons were active. And again, this stretched the definition of active to its breaking point, needing 1 spike event every 100 seconds, 10 times longer than the entire sequence of the task. So everywhere we've imaged, most neurons send no spikes in over a whole minute. So such imaging studies have repeatedly shown silent neurons in the cortex that left open many problems. Is there some technical problem with the fluorescent chemicals we used? Perhaps they did not respond to isolated spikes, and so made neurons look more silent than they were. Or did the chemicals not get taken up by all the neurons? In which case, the silent neurons could just be those that had no chemical inside them. Or did they get damaged by the chemical? In which case, the mere presence of the chemical alone caused neurons to stop spiking. And most imaging studies, including all of the above, look at neurons in layers 2 and 3, just at the top of the cortex, because it's harder for light to penetrate deeper, and thus for us to video deeper. Which leaves open the possibility that there's something special about these layers of cortex, and deeper neurons are all merrily spiking away. As in any area of science, each technology brings great insights, but just as many new potential drawbacks. But other teams then proved silent neurons existed using the phenomenally fiddly technique of patch clamping. Traditionally, neuroscientists just lower a sliver of metal or glass into the brain and pick up spikes when that sliver happens to be near a neuron's body. By contrast, patch clamping finds a neuron within an animal's brain by attempting to physically attach to it, patching to it. Because they are finding neurons by physical contact alone, the experimenters are not relying on activity. Patch clamping has its own biases. It is easier to patch a bigger neuron than a small one, and in a live animal, you still cannot see what you're doing, but crucially for us, activity is not one of them. Once attached, they can play the animal some sounds or get it to touch something and see if their attached neuron becomes active. Largely, it does not. Thomas Hromadka in Tony Zador's lab at Cold Spring Harbor patched a collection of neurons in the first bit of auditory cortex in awake rats and found most of them were silent most of the time. And silent regardless of whether the animal was sitting quietly or listening to an extremely dull collection of pure tones. Playing sounds to the bit of cortex that cares most about sounds evoked very little response. Dan O'Connor, then in Carol Saboda's lab at Genelia Farm, patched a collection of neurons in that specialized whisker bit of cortex in mice that were again using a whisker to find a vertical pole. And guess what? Most of those neurons were silent most of the time, even when the whisker was waving back and forth, hitting the pole. And both these and other studies have consistently found silent neurons in all layers of the cortex. But looking back, this epidemic of silence was there in plain sight. Theorists long ago worked out how many neurons should be within recording range of an electrode lowered into the cortex of a rodent. Simple physics says that the farther the distance between the electrode and a neuron, the weaker the spike signal will be. The strength of the signal should drop roughly exponentially, rapidly at first, then slowly, with increasing distance. And there will be some distance beyond which the signal will be too weak to detect with your equipment, because it will be indistinguishable from noise. So theorists imagine sticking an electrode into a collection of neurons packed as tightly as they are in cortex, worked out at what distance the spikes cannot be detected, and counted how many neurons sit within that distance. And the answer was at least 100. But when neuroscientists lower a single electrode input into position, they see at most a handful of spikes from different neurons. And we can work out they are from different neurons if the spikes are consistently different heights. They see nothing like 100 neurons, not even close. So all along that meant most neurons are silent. So the irony is dark neurons are essential to being able to record neurons in the cortex at all. If many of those 100 neurons were spiking, then tens of thousands of experiments would have failed. For the electrode would simply be awash with spikes, a ceaselessly fluctuating voltage from which individual spikes from individual neurons would be lost. Without being able to distinguish neurons, we cannot measure them, test them, work out what they like and do not like. So no Nobel Prize for Huebel and Weisel for the discovery of simple and complex cells in V1. No tuning cells in auditory cortex, no place cells in the hippocampus. Perversely, it turns out we have dark neurons to thank for our ability to make sense of the cortex. So, the long tail.
Awesome. Thank you, Mark. And, this is such an important point that you raised that, as you say, it's been kind of hiding in plain sight all these years, and it's become very obvious right now that we're recording from more and more neurons at the same time. We really can't avoid dealing with this fact. So I guess we wanted to first, in our questions, sort of address a bit of your process with this book, and then maybe we'll get in more deeply into the science of it. So we wondered, you've been sort of doing popular writing for a while, right, through your blog, and it's a pretty feisty blog, and we might chat more about that in a bit. But what took you into this particular book? How did you choose this topic to write about, and who would you say is sort of your target audience?
So so the gem of this idea came, I don't know, a few years ago. I remember I was standing on a freezing cold train platform waiting to go to work in Manchester. So I live in Sheffield, so I have to travel by train for hours. Used to have to travel by train for hours to go to work every winter morning, standing there freezing my stuff off. And I was idly thinking about whether it would be useful to consider the point of view of a spike when trying to understand both the transmission of a spike and the scale of the brain and what it would actually look like. And then it occurred to me that more than a scientific idea, that would be useful as a framing device for explaining lots of systems level neuroscience to a general audience who otherwise we could be completely incomprehensible to all this talk of calcium imaging and optogenetics and fluorescent dyes and so on. It kind of came together so I was approached by an agent about writing a book because they'd read some stuff on the blog and liked it and said, so I've got any book ideas. So I scratched my head a little bit and said, well, I've had this thing about what if we follow a spike through the brain? What if we take a journey with the spike? So actually put the reader with the spike itself and just follow it all the way through the brain so that you can see on the unfolding of the brain as you go and unfold all the facts about spikes. And then also on top of that unfold sort of the cutting edge of neuroscience that you never get to see reported in the sort of general media because it's considered to be too difficult basically to report. If it's not tied to a specific breakthrough in a drug or an animal model disease or something, you just don't see it. So he was very enthusiastic about that. So it got hammered into some sort of shape. And the book turned out pretty close to the original proposal, which was a surprise to me and him.
And so are you hoping that it will be read by young people coming into neuroscience as well, by neuroscientists, or really primarily aimed at people outside the field?
So yeah, I was being ambitious with this one. So I was trying to walk that tightrope where there was enough in it that someone who came without a deep background in neuroscience would be able to read it from end to end and get excited and really interested in it. So that the second reading would suddenly a whole bunch of stuff would click into places. You loop back around again. They go, oh right, I see where that comes from. But also I'm aware that often, particularly this kind of trade popular science book written for the public can, for the audiences a little bit more on the know, be a little bit kind of either pointing out the obvious or making endless sort of generalizations that don't really stack up. So I also wanted to make sure that it was a book that was as interesting to a practicing neuroscientist, keeping in mind, of course, that as we see at SFN every year, we used to at least, neuroscience is a huge discipline, and even most neuroscientists don't know a lot of stuff about how spikes work at the level of individual neurons to any degree of detail, because who has the time to read all this deep literature? Right? They need to work on the genetic stuff or molecular stuff or the receptor makeup or whatever they're working on, all that important stuff. So it's a chance to bring all this together. So it's kind of also a starter pack for your systems neuroscience view of the world.
I had a bit of funny discord just now when you read, because when I'm reading your book, I read it very breathlessly. Sort of it, like listening to you, there isn't that feeling of breathlessness. But when I read the book, there's a very clear contrast to, for example, Grace's book that we discussed last month. Grace writes this sort of, no pun intended, graceful narration of historic characters, where your style is much more, maybe natural born killers or Tarantino style writing, where we hop onto a spike and follow it and bounce around. And you talk much more about contemporary scientists that are not famous, that a popular science reader will not necessarily recognize. So did you do that on purpose? And how did you steer away from the superstars and more to the everyday neuroscientist, so to speak?
Partly from the desire to give a view of the cutting edge of what's currently known. So just by wanting to talk about the last 20, 25 years of systems neuroscience means naturally pulling on a bunch of names that are well known to us in our small audience, but not well known to the broader public, indeed. And I was making a conscious effort not to spend forever on the basics, not to spend just a whole chapter talking about how a spike is created. Just do that in 3 paragraphs at most, just because if you want to go deep into that, there's lots of stuff you can read about that. But to understand, all you need to really understand to understand the sort of spiking level of the brain is just roughly how they're created, so that you know why this thing is being sent, what makes it being sent and what doesn't. So that means that kind of compression of history means you could not then spend ages talking about the older superstars, as you say, the ones that are better known to the public.
Yeah.
So there's a few nods, of course, to, just mentioned Huben and Wiesel then. I mean, there are parts where it draws heavily on older data, but older data that isn't really discussed much anymore, like some of the classic cat visual deprivation experiments and that kind of thing. The kind of thing we also have to preface to the lecture going, we wouldn't do this now, but it was really useful that they did it.
But when you read it to yourself, do you read it in the tone that you read it to us now, or do you read it breathlessly and quick and action movie-like?
Yeah, I think it.
I had a totally different narrative voice in my head when I
dive back in to this next section, long tail, then I'll read it in more the voice in which it is written. So you're right. So the narrative style is clear. It was much more. It was intended to be a propulsive style, which carried a very strong momentum through it. So that even if there were bits where you didn't understand what was going on, you didn't really care because you would just be pushed to the next part, and just staying with the journey and staying with the sort of the ideas that are being flung your way. I think it's also partly just because I get bored quickly, right? So I don't want to spend like a whole chapter on a single thing. I find the greatest challenge is explaining really complex ideas as concisely and entertaining as possible. So having to explain, I just read through, having to explain calcium imaging in I think 5 sentences. That paragraph took a while, for example, to get that propulsive quality. You can get through it and understand exactly what it is, what it's for, what it does, and then move on.
I really like the way you weave in a lot of just sort of everyday pictures. You make it very visual, right, on the way through, which is great. I hadn't actually realized that you wrote this blog until we were sort of researching for today, and so I'd love to hear more about that. So how did you get into writing that? Who is your audience for that? It's pretty opinionated, which I think is something that people appreciate. So how do you kind of navigate the politics of opinions we were willing to put out there on the web?
That's a good question. Well, so I began it middle of 2016, I think. It was. I think I was looking for doing. I started thinking about doing more sort of public communication of science stuff. So I'd given a couple of public talks and they were okay, but I knew that I was better at writing than I was at speaking. So writing seemed to be obvious. So I for my own entertainment, wrote a few posts about papers that I'd read, posted them up, got a few reads, and then I wrote a post sometime in that summer about the Allen Brain Institute releasing their first massive data set of data, in which in a single sitting on the train on the way home, I think I wrote a whole 1,500 words, which was about sort of the conflict I felt about this data set being released, that it was amazing that it was being released before publication, but also that what it meant was that no one else would ever do this because that they can do this from the security of this private funding and knowing they've got jobs and so on, but who in their right mind would actually release data they've gathered without some sort of security that they're going to be acknowledged for it?
Yeah. Have you evolved in your thinking on that, by the way?
So it's still I mean, the Allen Institute is still a I mean, it's a terrific thing, right? And that and other sort of privately funded research, or the public-private ones, their ability to take that step away from the pressures of need to be all this immediate publication and immediate outputs and be measured constantly on that ground is great, and the fact that they can really look at a proper large scale, so you can really, really fine hone every part of the procedure. So you can have people who specialize in the testing equipment, people who specialize in the imaging equipment, people who specialize in the analysis. So that's all really, really put together more like CERN-like.
Yeah, and I think it actually also hits on your chapter, right, on dark neurons, because what that freedom gives them is to publish null results, right? That things turn out to be much more complicated than you realize. There are a bunch of neurons that just don't respond, and if it's your lab, you wouldn't publish that. You would just wait till the cool result came up. But they have the data, and everyone else has the data, and so they're able to acknowledge the full richness and sometimes boringness and complexity, right, that's in there.
Yes, that's indeed. Indeed, they're not beholden to the need to have the shiny new result come out, because it's just the report what is there. Which is cool. The only potential issue with something particular on the scale of the Allen Institute, of course, and we're probably going to see the same, I guess, with the International Brain Laboratory, is that there is such a huge scale that essentially they've cornered the market in that part of the brain and that kind of system, because it's difficult then for other people to be able to say, well, I want to work on this brain system, but I don't know what questions they're going to ask. So without knowing what questions they're going to ask, what am I going to work on?
I guess, I interact quite a bit with them, and we hear a lot more the opposite criticism is that you've locked yourself into this one thing, and really the thing we're interested in is this other thing. And the Allen people, we're stuck, we're not stuck, but, we've decided to do that, so that's too bad, right? So I wonder if that's, maybe grass is greener. So
yeah, pretty much.
I do know that a lot more about Janelia, actually, that, it's very tough, right, to run a fly lab when you're competing with a group that has all the resources as well as the nimbleness that they can jump on the coolest new thing, right?
Mark, maybe connected to this, but also going back a little bit to the book, it seems like your blog is at times much more snarky than the book ever is. So the book is sort of like a fest of positivity, whereas the blog may be written in a positive way, but it often has snark in the back door, or when you put it away, you're like, yeah, there was a little bit of spunk in there. So why did you keep that out of the book? Did your editor twist your arm, or?
No, actually. So no, there was no sort of editorial guide on that. I think it was just. I think a lot of the sort of more, I don't know, humorous asides and a little sort of joke jabbing is all about stuff that's happening in the moment. It's often about things that are happening right now. Whereas when you write a book, you want it to be something that in 10 years' time will still make sense and stand up. So because it's not a book about politics, it's going to hopefully age a little bit better.
Yeah,
it's not going to be like completely useless in 2 years' time. I mean, every book has been written about Trump in the last 4 years. It's just going to be historical interest only, really.
Have you received pushback for anything that you've ever written in the blog? And have you received any pushback for stuff that you've written in the book?
So,
like, have you gotten angry emails?
From the book? No, not yet. Obviously, slightly slower feedback timescale for the book. I'm assuming there will be at some point. But for the blog, occasionally, yeah. And only when emails and direct messages, and occasionally just live people writing whole threads on Twitter, did I realize that, as Adrian says a few minutes ago, do I realize that I've actually written stuff that was opinionated? So I often write this stuff and don't realize that it's like it's strongly opinionated, because I forget that that's kind of my writing style is quite I don't know,
pungent.
Yeah, propulsive, and it moves rapidly, and also talks directly to the reader in the way obviously I tend to use pronouns is often directly engaging rather than doing any sort of third person stuff. So I was so
interested in the way you framed the title of the book in terms of the journey of a spike, which sounds to theorists in general, I think, very feed forward, right? That there's a kind of direct line of transmission. But in fact, what your book does is unfold, I think, a pretty unique hypothesis or idea about what's really going on, right? That spontaneous activity is this key property of the brain that allows it to do all kinds of stuff. I was actually in grad school in Israel at the Weizmann Institute when Tal Koenig, did that beautiful experiment where she showed that using optical imaging that activity in visual cortex, spontaneous activity, kind of recapitulates patterns that you see in response to real visual stimuli. So I wondered if you would mind unpacking for us that kind of central thesis of your book a little more. You could go to the reading if you want, or and then talk about it.
Yeah, sure, okay. I could skip forward to
We
read a
can do
little
this
bit.
reading and then come back to that question if you like.
Okay, you want me to carry on with this chapter? I think we're going to ping into some of the more spontaneous stuff now.
Yeah. Great.
Yep. Cool. All right. Okay. Yeah, checking the background. Okay, and yeah, all right. I'll try and get it in the more. Now I've
Tarantino.
West. Yeah. Well, now I've got through the sort of the opening salvo. I can now relax a bit more and yeah, do a bit more like I'll pretend my kids are sitting in front of me. That'd be easy. All right. So the long tail. Wait. Yes. So when I say silent, what exactly do I mean? So we've been in your brain less than a second. When we make it to a full second, less than 10% of your cortical neurons will have fired a spike. In 1 second, there is 90% silence. If we were to hang out here for a whole minute, still the majority of cortical neurons would send no spikes. Yet, by tagging along with the spikes from that minority of active neurons, we have reached the farthest ends of the pathway through your visual regions of cortex in a few hundred milliseconds. So right at the top of the book, I told you there is an average of 1 spike per second for every neuron in the cortex. But if 90% of neurons are silent in 1 second, how can that be? If so many neurons are silent in a second, then to get an average of 1 spike per neuron, that must mean some neurons are sending loads of spikes per second. And they are. About 10% of cortical neurons produce half of all spikes. I'll repeat that because it took me a while to wrap my head around it when I first came across this fact. Half of all the spikes in your cortex are sent by just 10% of the neurons. In Hromádka's collection of neurons from the first auditory bit of cortex, 16% of the neurons contributed half of all recorded spikes. In O'Connor's collection of neurons from the specialized whisker bit of sensory cortex, exactly 10% of the neurons contributed half of all recorded spikes. These few are sending the majority of messages, dominating conversation like a minor bird in a monastery, which means there is a continuum with truly dark neurons at one extreme and these minor birds at the other. To find out exactly what this continuum looked like, in 2012, I surveyed data on the activity of groups of neurons across the cortex, part of my contribution to a mammoth review of neural activity in the cortex written with Adrian Wara and Christian Mackens. And everywhere, I found the same thing. When we choose a chunk of time to watch neurons, the number of spikes per neuron will be unevenly distributed. In that chunk of time, some neurons will be silent, most will send just a few spikes, and a handful of neurons will send many spikes. Distribution of activity across a group of cortical neurons is long-tailed. Let's show everyone the figure at this point now. Long tail. Yeah. Right. So I found this same long tail distribution everywhere. In the first regions of visual and auditory cortex, in motor regions, and in prefrontal cortex. In data using different methods of recording spikes, and regardless of what the animal was doing at the time, quietly sitting, seeing, moving, or deciding. Always, some silent, most quiet, a few yelling. My survey of silence has deep implications. For one thing, average activity is useless as a way of understanding what a region of the cortex is doing. That handful of loud neurons skews our averages upwards, a long way upward, suggesting to us that most neurons are sending spikes when they are not. For another, it showed that dark neurons, neurons sending far less than 1 spike per second, are everywhere. Most clearly, it showed that during the brief few seconds of an animal sitting, seeing, moving, or thinking, those dark neurons are not communicating anything to anyone. What then are these dark neurons for? What dark neurons are for? Dark neurons are a real pain. Our theories for how bits of the brain work are based on the patterns of spikes in them. But the dominance of dark neurons means that our theories of the brain are only about a mere handful of actual neurons. Remember the simple cells and complex cells in V1? When we were there, I rehearsed for you the theories of how the tuning of simple cells can be explained by combining spikes from their inputs from the retina, and in turn how the tuning of complex cells can be explained by combining the spikes coming from simple cells. Dark neurons make clear that these theories are but for a handful of neurons in V1, those cells that reliably respond when shown in pictures, and not for the masses that do not respond. Dark neurons mean that the emergence of brain-like tuning to the visual world in AI networks is not perhaps as insightful as it appears, as this brain-like tuning is a comparison to a relative handful of neurons. And these issues are not restricted to V1. They play out in all areas of cortex. Everywhere we have theories of single neurons, across the 2 visual highways, across the other senses, across the rest of the cortex. But these dark neurons must do something. The neurons are expensive to build, expensive to maintain, and expensive to use. Your brain uses about 20% of all your energy budget every day. Just keeping your brain cells alive and in good condition uses about 25% of the brain's energy budget. That's 5% of your total budget every day. We've already seen that synapses are expensive. About half the moment-to-moment energy used by neurons is in their inputs. The other half is in their spikes. Dark neurons burn energy to stay alive and burn more energy on their inputs, yet produce little to no output to show for it. Perhaps one explanation for them is to flip this argument, to think about the energy they aren't using. After all, one way to conserve some of the brain's energy budget is to not send spikes, for without them, you halve the energy cost of using a neuron. But if you didn't need the dark neurons, evolution wouldn't have brought them into existence. And development wouldn't have spent all that energy growing them, dividing them, and extending their axons to the right places. Your body has much better things to spend its energy budget on than dead weight in your head. It would be absurd to fill our visual cortices with neurons that do not see anything, absurd to grow a massive prefrontal cortex and then fill it with neurons that sat in darkness. So what are they for? So here are 3 ideas. The simplest idea is that in the lab, we're not asking the brain to do anything interesting. So after all, in the lab, we can only probe a tiny fraction of the real-world input these neurons receive. So maybe our experiments are just not rich enough, and we need recordings of neurons in animals behaving naturally over very long times, days, weeks, months, to find out what they are for. Then, if we sample enough of the animal's life, we can find out what the dark neurons respond to. Technically, this is just about within our grasp. Practically, less so. Some poor grad student has to actually do these recordings, locked in a lab for weeks on end, and lose their entire social life, their romantic partners, and their self-esteem in the process. But as Bruno Elshausen has argued, this dull world argument is plausible for V1. That the stimuli we use in our experiments are too simple, too poor a reflection of the real world, and all the neurons in V1 do respond to something in the world. It's just that we will never find out unless we record them for a whole lifetime. In this idea, the sparsity of activity is also about making the most out of what energy the brain has available. The theory is that V1 has so-called population sparseness, where each neuron in V1 is very selective to what it responds to, so the energy isn't wasted by a swathe of neurons responding to the same thing, sending redundant information. And this in turn means there is lifetime sparseness. If those selective things rarely occur, the neurons will rarely send spikes. In this idea, dark neurons are then the thought of our limited capacity to probe the brain, not the brain itself. A second idea is that the dark neurons are a reserve army, waiting to represent new things. The primate lifespan is long, packed end to end with things to remember, skills to acquire, faces to learn. And for humans, our unique ability to learn new concepts, ideas, and words places heavy demand on our ability to represent things using spikes. Some of that learning will be through changing the effective strength of connections between neurons. At the most extreme, through changing the strength from nothing to something, whether by increasing the size of the voltage blip, increasing the reliability of the synapse, or both. And increasing the effective strength of excitatory inputs to a dark neuron will in turn increase its rate of sending spikes. The upshot is the same as the first idea. The reason we do not know what dark neurons are for is that we track neurons only over the briefest moments in their entire lifespan, even in rodents that live but a few years, and so would not see if dark neurons are recruited over time. This simple idea is complicated by the fact that the brain can't just blithely increase the number of neurons sending spikes. For one thing, this will simply increase the energy drain. So the idea suggests that other neurons will need down-regulating as the reserve neurons are up-regulated. For another, these no spikes need to be balanced. An increase in excitatory spikes needs balancing by an increase in the counteracting inhibition to prevent runaway explosions of activity. A third idea is that the dark neurons are sending information just fine. They just do so jointly. Each dark neuron makes a tiny contribution, a single spike every now and again. But since dark neurons make up 90% of all neurons, that adds up to a lot of spikes. In this idea, the dark neurons send messages not by many spikes from a few neurons, but by many spikes from a massive group. And as an individual neuron needs to receive a legion of spikes to make a spike in turn, so this mass of dark neurons could be highly effective. What's more, not only are loud shouting neurons in the tiny minority, but also their shouting may well be tuned out by neurons on the receiving end. Remember, synaptic failure can act as a gain control, turning down the influence of shouting neurons and turning up the influence of quiet ones. Indeed, neurons whose inputs have short-term depression respond best to jumps in which of those inputs are active, not the total rate of inputs. So a group of dark neurons sending sporadic spikes together would be exactly the type of input that such a depression-equipped neuron was looking for. Synaptic failure could, paradoxically, favor neurons that rarely fire. A nuanced version of this idea also gives us a different view of the lifetime sparseness of neurons. Dark neurons are the vast majority in any region of the cortex. There are likely, then, many more with the capacity to communicate the same messages than are needed. So perhaps each response evoked from that region, like a picture shown to V1 or a sound played all through cortex, draws from a random subset of dark neurons. Random from our point of view, not the brain's. That random subset sends a spike or two, then shuts down. So, from our point of view, most neurons are silent most of the time, yet the message sent by the dark neuron population is the same each time. And this random recruitment idea has a simple experimental prediction. Record from a group of neurons while repeating the same event over and over again, showing the same picture to V1, or making the same movement of an arm, then most neurons should vary whether they respond or not on each repeat of the event, apparently at random. We see just this random participation in groups of whisker sensing neurons across hits on their whisker, groups of arm movement neurons across similar movements, and even in groups of crawling neurons across repeated bouts of crawling and sea slugs. Dark neurons are then not dark, just misunderstood. 3 ideas for what dark neurons are for, 3 ideas for solving the paradox of how neurons that send no spikes somehow contribute to the life of the brain. By contrast, those minority of neurons that reliably send spikes, those in the long tail, they should be easier to understand, right? No. Many of those seem to be talking without listening. Oh. So, talking without listening, final section of this chapter. So, hidden in the active neurons are many of another kind of dark neuron, a type 2 dark neuron. Neurons firing away just fine, spikes streaming out, but that do not seem to respond to anything, whose output of spikes does not meaningfully change no matter what is happening in the outside world. Talking to other neurons, but apparently not listening. Dark to the outside world. So these type 2 dark neurons were hiding in plain sight in huge piles of neuron recording papers from the 1960s through to the early 2000s. In those papers, the first sentence out of the blocks in the results was always something like, "We recorded a total of N neurons, one at a time, while presenting stimulus X when invoking movement Y. We found M out of the N neurons responding to the presentation and will be the subject of this paper." And always, the number M of responsive neurons was far less than the total number of recorded neurons N, and the rest were just thrown away. So what were those other neurons, the N minus M unresponsive neurons, doing? Type 2 dark neurons are especially clear in modern recordings of many neurons at the same time. But with these recordings, we can take huge samples of neurons, numbering in the hundreds or thousands, and examine each neuron individually for its tuning, or what elements of the world it responds to. When Simon Perron and colleagues looked at their recordings from that specialized bit of whisker cortex, they dubbed 67% of those neurons as active, and that included neurons firing barely at all. Searching for what the spikes from those 67% were responding to, they found 28% of them were sending spikes with no detectable task information at all. No tuning to whether the whisker was moving or hitting a pole, no apparent difference in whether the pole was in one position or another, or the trial was to lick left or lick right. 28% of the active neurons in the very first bit of cortex getting input from the one and only whisker being used were not apparently listening to what that whisker was saying. Christopher Harvey and colleagues found a similar story in their recordings from the parietal cortex of those mice running in a virtual reality maze. Remember, they had dubbed just 47% of their neurons as active, and of those, 27% were sending spikes with no task information at all. No tuning to where the mouse was or what it was doing. Active neurons in parietal cortex seemingly did not listen to what was happening to the world around the mouse. And my lab found the same thing in the prefrontal cortex, in recordings of neurons that came from rats running up and down a Y-shaped maze. In each session of training on the maze, our collaborators had recorded between 12 and 55 neurons, and all were active, or at least not dark, as these were electrode recordings. Yet, in each session, typically just 1 or 2 of the neurons responded differently to the things happening in the world around the rat. Only 1 or 2 sent different amounts of spikes, whether the rat chose the left or the right arm, whether the rat got a reward or not at the end of the arm it chose, whether or not the light was on at the end of the arm. It was like the active neurons in prefrontal cortex just didn't care about what was going on, but they very much did and how they did the story of the next chapter. You may be wondering, what do you mean exactly by respond? That innocuous question opens up a deep hole. Typically, it means that a neuron has sent a different number of spikes in one condition compared to another, like whether a whisker hits a pole or not, or whether the animal has gone left or gone right. And different is defined by setting a threshold, typically by asking whether the difference in the number of spikes between the two conditions passes some statistical test. This is how we end up with the ubiquitous idea that there are different functional types of neuron in the cortex. The neurons all over the cortex are either the type that respond to thing X, or they do not. So neurons are in V1 are either simple cells, or they are not. Neurons in auditory cortex are either tuned to a specific group of frequencies, or they are not. Neurons in somatosensory cortex are either tuned to the touch of a finger on the surface, or they are not. And neurons in motor bits of the cortex are either tuned to the speed of arm movement, or they are not. And neurons in prefrontal cortex either respond to the value of a reward, or they do not. But as Adrian Warrer, Christian Mackens, and I pointed out in 2013, using a dividing line to label neurons as either responding or not responding is a fallacy. It breaks into two groups the continuum of responses from the non-existent through the weak, the moderate, the middling, to the exuberant. We can always move the line and break the continuum into two different groups. And this means there are not really types of neurons we can define by their responses to the world. We can reel off many examples where apparent neuron types do not exist, where neurons sit on a continuum for how they change their stream of spikes. We've already seen for V1 that simple and complex like cells sit on a continuum. In the prefrontal cortex of monkeys touching a vibrating bit of metal, there is a continuum for how much the neurons there respond to the vibration's frequency. In the parietal cortex of rats making left or right decisions based on the number of clicks or flashes, both the neurons' preferences for direction and their preference between clicks and flashes lie on a continuum. Which all means type 2 darkness is one end of a continuum of responsiveness. What we've been calling type 2 neurons are those that barely change their spiking to an event in the outside world, a change too weak to be picked up by our analyses. But are the changes too weak to be picked up by other neurons in the brain? After all, what if these weak changes in the stream of spikes happen in many neurons at the same time? To answer this properly, it's time to turn to the deepest question in neuroscience, the meaning of spike.
Thank you very much, Mark. Thank you. I think maybe I invited some of the people that asked questions onto the screen. I don't see them accepting. Yuri and Mira, John, if you would like to come online to ask your questions, that would be lovely. Yuri, I can also tell you, I just see you rejected my invitation. I can tell you the happy news that you've won Mark's book, but it's okay. Mark will send you a signed copy with a nasty note that you didn't come online to ask your question. But what Yuri wanted to ask you was what the failure of spiking, how the failure of spiking plays into all of this, whether that is a bug or a feature in the neural code, and how that connects with the dark matter of the brain.
Yeah,
you've mentioned it in the discussion before, so I sort of missed my window to ask it at the proper time, but maybe you can come back to it.
Sure. So the entire previous chapter of the book to this one deals with synaptic failure, and I deal with the idea of dark neurons and synaptic failure separately. So the dark neuron chapter is really about pointing out how much of the brain there exists that we have very little grasp of what it's doing. Synaptic failure one is all really about the idea that it starts by posing that the synaptic failure seems to be a bug, right? Why would you, again, it's a problem motivated by energy requirements. Why would you spend all this unbelievable amount of energy generating spikes, only for them, when they get to the synapse, to them to not cause any release of transmitter at all? So their signal just is lost to the neuron on the other side. And those failure rates can be astonishing. And so in various recordings in hippocampus, individual synapses can fail up to 90% of the time, which means only 1 in 10 spikes is making anything happen when they arrive there, which is an atrocious waste of energy. And yet we know that in other parts of the brain that the synapses can be 100% reliable. So there are synapses in between interneurons in the striatum which are 100% reliable. So that means that it appears that failure can be as much a choice the brain makes as something that's an inevitable consequence of the noisiness of biology.
You mentioned somewhere else also, right, that it's part of the strengthening process of a synapse may not only be a change in efficacy in terms of the impact on the postsynaptic membrane potential, but also simply tuning its reliability as part of the plasticity process. At least you say that in an earlier chapter.
Yeah, right. So so I speculate that so one of the sort of more immediate reasons why we have synaptic failure is to provide some ability to tune weights that is independent of the size of the voltage response on the other side, so that you can over time the effective weight is a product of the failure rate and the size of the voltage response, which we sort of hide in most of our models as just this is the weight, where actually it's easily, even at that level, two separable components, and then you get down into the synaptic level and it's more. Also in the book, then I take the reader through a whole bunch of theories about why synaptic failure exists. And one of, I think my favorite of those is the idea that it's there to throttle information on purpose. And it's so that it's the idea that if you look at the axon of a neuron, it has a finite and pretty small rate of information that it can sustain, given an average spiking rate in cortex. And just as I argued, talking about for the dark neurons arithmetic, a neuron gets far too many spikes than it needs to make its output spike train. So that means it's getting far more information in than it can possibly make use of. So one reason for synaptic failure from this point of view is deliberately to turn off lots of information coming in. So the rate of information coming in matches the rate of information going out. So you have you keep the neuron that is in an optimal state. And that means that what it predicts is that what's controlling the failure rate should be the postsynaptic neuron, that it's controlling the failure rate of its own synapses. Indeed, a whole bunch of studies from Tiago Branco seem to show this happens. You stimulate locally a tiny bit of dendrite, and you can, to increase the efficacy of a couple of synapses from a single neuron, and the failure rate starts to go up as a consequence.
Niranjan, I'm sure I butchered your name, but thank you very much for coming on screen. Please feel free to ask your question.
No problem. Thank you so much for giving me the opportunity to ask Mark a question. Mark, first of all, a great book. Thank you so much for the book. And I've just ordered it, so I'm hoping after listening to you, I wanted to get the book. So,
cool.
So one thing that I wanted to ask you, that this whole theory of memory and learning is based on synaptic plasticity and that how this network of neurons are active in a particular configuration, and that is basically the representation of memory, they call it. But listening to you and to some others who have basically talked about it, I think Laughlin's book, where they talk about how expensive it is to actually maintain spikes even in a single neuron for a given amount of time. So I wanted to get your opinion of what is it actually the function of or the purpose of spikes is. Does it actually, or does it involved in only transfer of information from one neuron to another neuron, or is it actually, do you believe that there are these network of neurons which actually give rise to represent certain aspect of environment or memory?
That's I guess the full answer to that question would be an entire book, right? But I guess what you're getting at is are you asking really about sort of about sustaining representations of memory and the environment through sort of particularly? Yeah. So I know, for example, there's a lot of obviously the heated debate currently in the prefrontal cortex literature about what's sustaining working memory, whether it's the classic papers showing the Goldman Rackage papers showing you have these individual neurons which are apparently firing throughout a long delay period after a monkey has seen a light at a particular position. Light goes off, you see these neurons spiking away. That seems to argue for a constant spiking model of working memory, and there are many computational models that realize this idea. But there's been pushback against that, partly because of these reasons of, is it really really realistic that you have these thousands of neurons of prefrontal cortex firing at 30 spikes per second for two seconds or up to a minute, because that's a huge number of spikes. And when you go and do record large populations, then people like Earl Miller report that what you see more of course, is that there are more of a relay race, that individual neurons fire little bursts, and that sequence repeats. And that sequencing of individual spikes is actually the memory, not these tonic neurons. Contrary to that, of course, we do know there are neurons in the brain that do fire these constant activity all the time. So there's neurons in the entorhinal cortex that can fire happily sitting in a dish without any input whatsoever. They'll fire away at 10 hertz. So
I was wondering. Sorry to interrupt you. I was wondering about long-term memories, right? In terms of memories of a lifetime. I mean, if I ask you something about your childhood or your major event of your life, or when you first got the book ready, manuscript ready, or something like that, you have a clear, vivid memory of it. So it must be stored somewhere in the brain, and the whole theory of it is stored in terms of spiking or synaptic plasticity. So what do you think of that?
So I'm gonna give the politicians answer that one. So I'm gonna duck that question. I would say that we have an extraordinarily poor grasp of what long-term memory is. We have some excellent models of relatively short-term memory storage, right? So skill learning changes synaptic plasticity. We can observe that in motor cortex. We can play with it and disrupt the skill learning. That's fine. But memories of long-term episodic stuff, I would say we don't really have a good grasp of what it is. Not least because we have the problems of things like the fact that the turnover of spines and synaptic strength in cortex is really quite rapid. So you can observe neurons in motor cortex have turnover of spines every 2 weeks. So what if there is some sort of long-term memory being sustained by these connections? There is some stuff we don't know about that's keeping this going. So before we even get to the sort of level of how spikes are a long-term memory, we would need to know a bit more about how they're stored at all to know how that then becomes activity.
Right. Thank you.
We should move on to the next question. Thank you so much for your question. We have another one, and I don't think, is Panos going to join us online or not, Tim? So I'll just read it up.
Yeah, I think read it out.
Yeah, so he says, the scope of the book is grand and the tone assertive. How would you escape the fear of erring in terms of scientific narrative? I would assume it poses a major block for any aspiring science communication author.
Yeah, you have to get happy with being wrong, unfortunately. So, yeah, I mean, I was lucky enough.
Blog post about that. You should. What did you say in that blog post? That most of your work is wrong, and you're fine with that.
Yeah, yeah, that's right. Yeah, that's my little take on the replication crisis, reproducibility crisis. Was going. I expect most of my work to be wrong. I'm a theorist, so it's so perhaps that's a more that's the attitude. Depends on what area of science you come from. I mean, I was lucky enough. So when I was doing my PhD in my early postdocs, I had Pete Redgrave as one of my mentors, and his line was always was that he was more happy to be wrong than right. That it meant that he was creating lots of ideas. Lots of people would want to then attack and address those ideas and show they were wrong, and then stuff would, science would move forward. So that was fine. It's also an inevitable thing of doing science communication that you inevitably will be accused of missing out points and nuances and simplifying complex issues and so on, because you have to, because that's the whole point of science communication, is to try and get across the general idea while removing some of the craft. But again, in part, the training as a modeler really helped there, because you have to spend your entire modeling career trying to remove all the extraneous biological detail to get down to the thing you believe is the true essence of what the system is doing, and then get told by all and sundry that those details are really important, and you have to, and so on. So in that respect, excellent scientific career training for this science communication. But yeah, I've no doubt I have erred frequently in the book, but to the best of my knowledge, I haven't.
Mark,
Okay, we'll ask you one final question. and we'll let people go. Maybe Tim framed this, and I think it's one we'd love to hear from you. So, is the brain a computer?
Wrong answers only.
Which answer? All right, so I'll get the standard answer. They're going, that's okay. So frequently when people ask that question, I mean, I know, not you guys, but frequently when people ask that question in the general public sphere, they're asking a question about a metaphor or an analogy, right? And it's not a metaphor or analogy, it's a scientific question. It's given the definition of a computer, does the brain fulfill that definition? And if you take a really hardcore computer science stance on what the definition of a computer is, a computer is something that executes algorithms. An algorithm is something that can be executed on a Turing machine, then the brain is a computer probably, because an algorithm is such a general thing to do that you can argue that a rock implements a particular algorithm, an algorithm according to the particular laws of physics that it obeys. So there is a kind of a trivial sense in which the brain is a computer, but there's not a particularly interesting sense. An interesting sense would be, can the brain be a Turing complete computer, which is, can it implement everything a Turing machine can do, and therefore implement all the set of algorithms that you need to do any general computation. Right? That's a more open question. So there are people like I was going to say Blake Richards, who would believe that yes is a Turing complete. I keep with his name. I keep trying to complete a Blake Edwards because the director, Blake Richards.
He's not here tonight. I checked with him.
Okay, good. Ask that question besides just taking over. Yeah. But there's another sort of interesting question. Is that almost, as far as I know, all our models of computation that are based on algorithms, they're all based on discrete steps, and the brain doesn't work in discrete steps. It has it's a continuous time system, well, down to the Planck scale, but it's a system that works within a continuous, turns this weird continuous binary switching. And we don't, as far as I know, have any formal models of computation that address everything that the brain does. So in that respect, we don't know, does the brain commit to a particular formal model of computation? I mean, I know that a long time ago, Wolfgang Mass was working on these ideas, about third models of computation and so on, which gone a bit quiet, presumably because it's really, really hard. But yeah.
Good. All right. Adrian, do you want to say the final goodbye? No? All right, Mark. Then it's on me to say thank you very much for coming tonight. We left a few questions that were of more technical nature out because they didn't quite fit at the end. For those of you who want these questions, ask them on Twitter. I'm sure Mark will answer. There's another ongoing debate tonight whether the brain performs gradient descent, so also feel free to flip into that one. And with that, Mark, I think we should let you go. Thank you very much again for coming. Very nice. I hope I get to write a book like yours one day when I'm old and wise like you. And until then, thanks again and see you next time.
And thanks to our audience for showing up today.
Yeah. Okay. Thanks, guys. Thanks for the invitation. And thanks, everyone, for listening. Thank you very much. Okay. Good night, all.
Good night.